A review on eco-friendly formulation of silver. Nanoparticles and its activity. Abstract: silver had been identified as a harmless, safe inorganic compound and had been used as an antibacterial/antifungal agent over decades. Silver had extensive application in various domains including pharmaceutical and chemical industries, especially in nanoparticle form due to its efficient activity. The size of the nanoparticles varies from 1-100 nm. There is numerous method available for the formulation of nanoparticles, but they are not convenient and economic, and also the reactives used for formulation of silver nanoparticles can cause ecological impairment. Presently the researchers had a thirst in this area by a formulation of nanoparticles from naturally available resources in a most effective manner and also is an economic way. This article precise the various green-mediated formulation of silver nanoparticles and their morphology, characterization, anti-microbial activity. Keywords: harmless, nanoparticle, green-mediated, anti-microbial, economic, morphology, characterization. Introduction. The term nanotechnology is characterized as structure creation, characterization, and application by changing its size and shape. One commonly, the size of the nanoparticles ranges from 10-100 nm in diameter. It is broadly classified into two types as natural and inorganic nanoparticles. The nanoparticles silver, gold, titanium, zinc, and copper are grouped into organic metals, and the carbon nanotubes and quantum dots are grouped into inorganic metals. Among these various nanoparticles, silver nanoparticle, N, had a unique attraction due to its metallic and biochemical activity. N also used in various applications including pharma industry, biotechnological, and food industry in versatile research approaches. During centuries, before the evidence of penicillin activity, silver had been used as a therapeutic agent. As the silver comes in nanosize, less than 100, due to their size reduction it shows considerable activity against different microorganisms besides gram-positive and gram-negative bacteria. In recent years the silver nanoparticles also had a great focus in the agricultural field. It was used as a fertilizer as well as pesticides. Due to their nano size, the particles can easily pass through the cuticle of the plant and showed a controlled realized activity. There are diverse methods available for the formulation of nanoparticles: physical, chemical, microbial. But they are not cost-effective, and also the chemicals and microbes used for the formulation of silver nanoparticles cause harm to the ecosystem. In the development of the field of nanotechnology, the researcher had used various naturally available resources for the formulation of nanoparticles, for example, oil cake, leaf extract, fruit extract acts as a reduction agent and capping agent. The nanoparticles synthesized by thesis methods show a prominent activity, and also it becomes cost-effective compared to other chemical and microbial formulation. The researchers reported that the naturally available phytochemical compounds, for example alkaloids, flavonoid, terpenoids, saponin, etc., present in the extract act as a reduction agent that reduces the metallic silver into a metal nanoparticle. Silver nanoparticles also used for biofilm formation and in wastewater treatment for heavy metals removal, and also for the microorganism trapping to purify the water for various utilizing activities. This review focuses on the various green-mediated formulation of and their biochemical activities and their characterization technique. Various green formulation methods. Green formulation of AN using Azadirachta indica. The leaf extract of Azadirachta indica, in Indian language is called neem, is used for the environment-friendly formulation of nanoparticles too. The researcher had 2,3 reported that the high amount of flavonoid and terpenoids compounds presence in leaf extract act as a reducing agent that reduces the Ag and ion into AN particles. The researcher mixed the leaf extract and 1 mm AgNO3 in different concentrations at test tubes by brown color formation. The formulation of nanoparticle had identified and observed OD at 200-800 nm3. The analyzer reported that the very high concentration of leaf extract is not suitable for nanoparticle formulation and identified that 1/3 proportion of sample and the AgNO3 showed prominent antibacterial activity against E. coli and other gram-positive bacteria with zone formation of 20.L8.66 mm, 50.L18.66 mm, 100.L22 nm. Green formulation of AN using Piper longum, long pepper. AN synthesized by using Piper longum displayed extreme cytotoxic activity against the Hep2 cell line 4. 90 ml of 1 mm aqueous solution of AgNO3 and 3 ml of leaf extract was added. The mixer kept in dark and maintained by room temperature. The formation of yellow color suggested the emergence of silver nanoparticles and the absorbance was estimated at 200 to 600 nm. The nanoparticles were produced at various sizes 17.6 nm, 29.3 nm, 31.6 nm, 37.5 nm depending upon their sample and AgNO3 mixing proportions. N3 identified that the notable cytotoxic effect was showed in 500 g/ml concentration of AN in that 51% of the Hep2 cell line were dyed. Green formulation of AN using leaf extract of Spartium junceum. The Spartium junceum solution used for nanoparticle formulation 5. And the researcher proceeded to 5 ml of 1 mm AgNO3 was added into 25 ml of S.junceum solution. The mixture was heated for 24 hours at 800 C. By the observation of color changes from yellow to brown indicated the existence of silver nanoparticles 6. The morphological characters were analyzed by SPR, UV spectrophotometer, and FTIR7. Green formulation of AN using Impatiens balsamea and Lantana camara. The leaf extract of Impatiens balsamea and Lantana camara used as a reduction agent for the formulation of AN 8. Different concentrations of AgNO3 stock solution were prepared, 1 mm, 2 mm, 3 mm, and the leaf extract of I.balsamina and L.canara was added to it and the solution was heated at 600 C for 5 hours. The nanoparticle formulation indicates the observation color change 9. The researcher identified that the nanoparticles synthesized by these leaf extracts showed high anti-microbial activity with the zone of inhibition from 11.03 mm to 13.08 mm 10,11. Green formulation of AN using Amaranthus viridis. Amaranthus viridis, cosmopolitan species commonly called as slender amaranth, in Indian language. Leaves and seeds were edible and are used as vegetables and tops. They are high in calcium, iron, and found to be bone of vitamin B and vitamin C sources. It also showed anti-diabetic and anti-hyperlipidemic activity 12. Amaranthus viridis had used for large-scale production of nanoparticle formulation had been reported. Leaves were collected, washed thrice in distilled water then washed with milli-Q water to remove water. Leaves were dried in room temperature for about 2h. Then 5 grams of leaves were chopped and boiled with milli-Q water for 50 degrees Celsius at 10 minutes, then extract using Wattmann No. 1 filter paper. 1 mm of AgNO3, 0.0421 grams, dissolved in 250 ml of milli-Q water in the orangey-colored bottle. AN were synthesized by dissolving 5 ml of leaf extract in 20 ml of 1 mm of AgNO3 and heated at 100 degrees Celsius. The color turns light yellow to reddish-brown, confirms AN present in the extract. Synthesized AN were characterized under FTIR, shows band range of 3423-2850 nm, scanning electron microscopy, energy dispersive spectroscopy, SEM-EDX, transmission, and electron microscopy which measures the size of the band ranges from 5-20 nm, XRD13. The stability of reduced AN analyzed by UV visible spectroscopy, peak observed at 4235 nm. As compared to other sources, Amaranthus viridis highly effective and useful, antibacterial activity against gram-positive and gram-negative, and from the Amaranthus viridis contains oxides, calcium, silver 14. Green formulation of AN from Ceratonia siliqua, carob. Simply known as 2 minutes reaction time at lukewarm for reduction of Ag and ions to AN. Leaves were washed with distilled water, dried, sliced into small pieces and boiled in distilled water for about 10 minutes, filtered the extract which was in yellow, then centrifuged to remove bulk biomaterials at 1200 rpm for about 5 minutes. 15 AN were synthesized by dissolving 5 ml of extract in 1 mm of 100 ml of AgNO3. The mixture changes into black light which confirms AN. By comparing to other carob plants, Ceratonia siliqua shows a quickly synthesized 1. The concentration of AgNO3 and leaf extract were changed from 1 to 4 mm and 5% 10% by volume. The characteristics analysis of AN involves UV visible spectroscopy, shows strong resonance band of surface plasmin at 420 nm16 which indicated the formation of AN. FTIR measures the band at 4000 to 400 cm1, SPR produced a peak near 420 nm, XRD founds the crystalline size of AN around 18 nm which shows that the AN are nanocrystalline. SEM measures the particle size ranges from 5-40 nm. AN could be controlled to 5-40 nm by varying the extract volume, and the AgNO3 leaf concentration were used as a reduction agent and highly effective against E. coli 17. Green formulation of AN using Cynara scolymus artichoke. Species of cyclamen, cultivated as a part of the food, leaf extract were known to show antioxidant, anti-inflammatory, anti-allergic, anti-ulcerogenic, and anti-hepatocellular carcinoma activity, and they are abundant in metabolites such as chlorogenic acid, luteolin, apigenin derivatives of kynarin acid and flavonoids. Leaves were collected, washed thrice in deionized water, chopped into small pieces. 200 gm of leaves were added in 400 ml of deionized water were added to 1 L of Erlenmeyer flask heated to 100 degrees Celsius for 2H, then filtered using Wattmann filtered paper. 10 ml of 20 ml of AgNO3 in 100 ml were added to 20 ml of extract in a dropwise manner. Color changes brown to orange. The shreds of evidence shows the formation of AN. 18. The characteristic analysis of AN includes UV-vis spectroscopy measures the particle size in the range of 200-800 nm, FTIR analysis the size ranges from 400-450 nm, SEM measures the particle size of the 200-223 nm and was 98.47 plus or minus 2.04 nm, and the zeta potential measures the particle size at 32.3 plus or minus 0.8 mV, and able to analysis the particle size less than 100 nm. 19. Green formulation of AN from aloe vera. Aloe vera is commonly known as a medicinal agent including anti-inflammatory activity, UV protection, anti-arthritis, antibacterial effect, and promotes wound and burn to heal. Properties. Constituents include lignin, hemicellulose, and pectin, which were used as reducing agent. 25 gm of aloe vera washed with deionized water, chopped and boiled in 50 ml of deionized water for 20 mins. The extract was filtered. 0.3 mol of AgNO3 mixed in 20 ml of deionized water with 20 ml of extract and stirred vigorously at room temperature for 30 mins. Nanoparticles produced at 6H, size ranges from 70.70 plus or minus 23, 79.47 plus or minus 22 and 161.66 plus or minus 530 nm, prepared at 100 degrees Celsius, 150 degrees Celsius, and 200 degrees Celsius respectively. At 12H the size is 95.25 plus or minus 23, 149.55 plus or minus 47 and 92.02 plus or minus 53 nm at the same temperature maintained in 6H. The leaves were effective against gram-positive, Streptococcus epidermidis, and gram-positive, Pseudomonas aeruginosa. Hydroquinones act as reduction agent. 21. Green formulation of AN from Ocimum sanctum, Tulsi. Tulsi was used in the medical industry, found to be a great use. 22. Formulation of nanoparticles involves the collection of leaves, washed in distilled water, cut into fine pieces. These pieces were added in 100 ml of distilled water and stirred at 60 degrees Celsius for 1H. Extracts were then filtered with Wattmann filter paper. 5 ml of extract solution dissolved in 5 ml of 1 mm of AgNO3. Nanoparticles were brought under characteristic analysis. UV-vis spectroscopy measures between 250 and 800 nm ranges. Surface plasmin resonance of particles found to be the peak at 413 nm and an absorbance band produced at 260 nm. TEM involves the analysis of particles in the range from 4-40 nm with a diameter of about 14.32 plus or minus 2.5 nm. XRD estimated the size of 15 nm. 23. Green formulation of AN using Rosa rugosa, beech rose. Rosa rugosa, commonly used as an ornamental plant, medicinal, cosmetic, aromatherapy properties. Preparation of nanoparticles procedure involves leaves collection, washed with distilled water, 50 gm leaves in 500 ml Erlenmeyer flask and boiled in 250 ml ultra-pure water for about 20 minutes. For silver nanoparticles, formulation 60 ml solution in 1 mm AgNO3 and added with 2.5 ml of leaf extract. The color changes from reddish yellow to deep red showed the confirmation of AN formation. Formulation did within 10 mins 24,25. Extracts were used for herbal medicines and vitamin products. An analysis involves UV vis spectroscopy, measures the size ranges from 300-1000 nm and diameter ranges from 1035 nm. Other methods for analysis included as FTIR, TEM. The absorbance increases with more leaf extract concentrations, and found to be more stable at the pH from 5 to 1026. Green formulation of AN from Shorea tumbugia. Shorea tumbugia plant extracts extremely used for earaches and outer shots in the arm. 27. The stem was dried for 10 days, kept in a hot air oven at 60 degrees Celsius for about 24-48 HRS, then crushed into a fine powder, 1 mm of AgNO3 dissolved in plant extract and make up to 200 ml, then centrifuged at 18,000 rpm for 25 mins. Stem bark was act as the reducing agent. The color changes from dark yellowish to brown and indicates an existence. 28. The analysis involves UV vis spectroscopy did by UV 2450 and measures the peak at 430 nm. SEM did by Hitachi S4500 SEM machine for image acquisition. It showed the spherical shape and diameter of about 40 nm. EDAX used for the analysis of weight percentage of AN found to be 33.52%. 30. Green formulation of AN from tea leaf extract. The reaction was carried out for 1-2H at room temperature is reported. Here tea leaf extract acted as a reduction agent. Nanoparticles were formulation by the following procedure involves of 16 grams of dried tea leaves were added in 100 ml of ultra-pure water in 250 ml of Erlenmeyer flask, boiled for 5 mins, 70 L of AgNO3, 10 mm, were mixed with 14.25 ml of tea extract in a drop-wise manner and followed by vigorous stirring. The color changes from brown color indicates the existence of AN. Formulation AN were characterized by TEM, XRD, FTIR, thermogravimetric analyzer, zeta potential analyzer. Synthesized nanoparticles were found to be spherical and range in size from 20-90 nm, shows the activity against E. coli. AN were produced at the competence of 99.1%, 99.7%, 99.9%, 99.8%, 94.6% and 95.3% with 5%, 10%, 25%, 50% and 100%, V/V, T extract respectively. 31 FTIR shows that the synthesized AN had the involvement of amides, carboxyl, amino group, and polyphenol. And also synthesized AN were discovered as low as compared to the PVA-coated AN, uncoated, commercial AN, which have good stability due to their functional group of the tea extract. Synthesized nanoparticles show the antibacterial activity against E. coli. 32. Green formulation of AN using Tectona grandis. Tectona grandis, the bark had been used as an antibacterial compound 5-hydroxy-1,4-naphthalene-dione, juglor, shows antibacterial activity against Listeria monocytogenes and methicillin resistant to Staphylococcus aureus, MRSA. 33. And used for the treatment of anemia. 34. Reducing agents in the plant constituents like carbohydrates, fats, enzyme, flavonoids, terpenoids, polybenzols, and alkaloids. Seeds had collected, before washing the outer coating was stripped and cleaned, dried at ambient temperature for about 3 to 4 days, then crushed into fine powders. 5 grams of powdered seeds were taken in an Erlenmeyer flask of 250 ml and it was added with 50 ml of double distilled water, boiling at 80 degrees Celsius for 15-20 minutes. Extract was filtered using muslin cloth, 1 mm of AgNO3 in double distilled water were prepared at the ratio of 1:4 and mixed below to boiling point, and the reaction conducted in dark place with the help of magnetic stirrer at 800 rpm. The extract turned colorless to yellow to reddish-brown color within 1H. Analyzed by UV-vis spectrophotometer and scanned ranges from 300-600 nm. Measures the maximum absorbance at 440 nm35. It showed the formation of nanoparticles. In some the images were blurred and could not able to take image at 100 nm of particles, therefore, FESM were did. The FESM EDX indicates 94% of Ag and 6% of oxides. TEM analysis at the ranges of 1030 nm36. Green formulation of AN from tomato leaf. Leaves of rosemary, Rosmarinus officinalis, were collected and washed with sterile distilled water. 5 grams of leaves were added to 100 ml of distilled water and boiled for 5 minutes. The extracts are filtered with Wattmann filter paper and centrifuged for about 5 minutes at 4000 rpm. The supernatant used for formulation of AN, 1 mm of AgNO3 added in a drop-wise manner in 50 ml of extract and incubated for 18H at room temperature, centrifuged 10 mins at 10,000 rpm. The color changes from yellowish-brown to brown after 24H of incubation indicates the formation of nanoparticles. The synthesized nanoparticles were analyzed under UV-vis spectroscopy, which measures the particle size at 300-400 nm in a resolution of 1 nm. Absorbance band located around 4 to 7 nm. Leaf extract acted as the reducing agent. Synthesized nanoparticles harmed wheat seedling and vegetable growth of tomato and wheat plants and enhanced the activity of peroxidase and MDA37. Green formulation of AN using capsicum frutescens. Capsicum peppers are produced. Capsaicin is ubiquitous. The capsaicinoid alkaloids were responsible for pungency. The leaf extract reduces as well as stabilizes the Ag and ions, and which contains secondary metabolites. The crystalline form of silver nanoparticle was analyzed by XRD. The bactericidal susceptibility and antibacterial activity are achieved by the method of disc diffusion for the synthesized nanoparticles. The resulting plates observed for E. coli, which indicates gram-negative strain, and observed B. subtilis, which indicates gram-positive strain. After they incubated the plates at 370 C for 24H. The diameter of the area around the well was analyzed for antibacterial activity. SEM observed the size of the N, with a range of 15-20 nm. The antibacterial activity mechanism was electrostatic interactions and cell wall disruption. The synthesized AN high activity against gram-negative and moderate against gram-positive 38. Green formulation of AN using Clitoria ternata and Solanum nigrum. The silver nanoparticles activity contains many materials used in medicine and in burn treatment and arthroplasty to reduce infections. It also prevents bacteria colonization on prostheses, catheters, vascular grafts, dental materials, stainless steel materials, and human skin. The Solanum nigrum extract as a reduction agent which is responsible for infant mortality. The extract was added with 1 mm AgNO3 and it was centrifuged. Then the pellet was taken and washed thrice with 5 ml distilled water and it analyzed by FTIR. The enzymes and proteins are not capping with N. The range of absorbance was measured at 420-440 nm. Clitoria ternata and Solanum nigrum silver nanoparticles stood against the pathogen's growth. 39. Green formulation of AN using leaf extracts of Pimenta dioica, allspice. The Pimenta dioica, allspice, leaf extract was resistant to spoilage. The lifespan of the extract was until 3 months. The leaf extract of Pimenta dioica has antiseptic, anesthetic, and other medical properties. Several researchers have reported no beneficial results from sun-dried leaves. The extract acts as a reduction agent that is mixed with AgNO3. The synthesized nanoparticles were analyzed by UV spectrophotometer and AFM. The NanoSurf Easy Scan 2 software was used to analyze the AN from the leaf extract. The largest size of AN from fresh leaves in the ratio of 1:1 and the smallest size of silver nanoparticles from dried leaves 40. Green formulation of AN by an oak leaf and fruit extracts, Quercus. The author had reported that the oak leaf extract and its fruit extract had been collected and washed with distilled water to remove the unwanted particles and sun-dried for 2 days. The dried matter was crushed with mortar and 3 grams of ground fruit was boiled with 100 ml distilled water for 10 minutes and 60 degrees Celsius. The blend of leaves and fruits was filtered using Wattman No. 1 filter paper and proceed in dark at 4 degrees Celsius. The 1 ml filtrate was added with 30 ml of 1 mm AgNO3. After 80 minutes the resulting color appeared in deep brown, which indicates the formulation of nanoparticles than leaf extract. The pH was adjusted using NH3PO4 and NaOH. The synthesized solution was observed by UV spectrophotometer. It showed an absorbance peak at 415.445 nm. Additionally, the solution was centrifuged for 5 minutes at 15,000 rpm. Light yellow to brown for leaf extract and pale brown to dark brown for fruit extract depending on concentration. It showed that silver nitrate reduction to silver nanoparticles. 40. Conclusion. Based on the above discourse, it can be stated that the formulation of silver nanoparticles paved the way for biomedical nanotechnology for developing effectual antimicrobial drugs in future perspective. By the emergence of green formulation, it largely scaled up silver nanoparticle formulation over physical and chemical methods in a cost-effective and eco-friendly manner. The morphology, characteristics and activity of AN varies according to their reaction parameters. This review provides extensive knowledge of the green-mediated formulation of silver nanoparticles being introduced over the last 10 years.